Trends over the last 16 years during the molting period have estimated a dramatic increase in the number of seals utilizing Long Island's waters. However, these trends do not provide a concise understanding of species composition, haul-out patterns, and the movement of seals needed for effective conservation and management. Tagging can be used for harbor seals, but it is too invasive and disturbs the environment of the seals. The use of photo identification with human assistance has great potential to utilize pelage patterns on seals for identification of seal populations, and it is less invasive than tags. How can we use photo identification as a tool for identifying harbor seals and their parameters? Seals belong to a group of mammals called pinnipeds. Within this group, there is a subgroup called Phocidae, which contain true seals. All 5 species of true seals are native to Long Island. In this group of true seals, there is the harbor seal. Harbor seals often lie with their heads and hind flippers elevated in what is often referred to as the banana-like position. This can be indicated with the yellow line. Harbor seals possess varied pelage patterns, usually on the ventral portion of the seal. These distinct pelage patterns can be input into photo identification softwares and can be used as a useful method for identifying seals since they vary between each organism. Several softwares have been proven effective for identifying different animals. Sci Manta is a program that can be used to identify organisms. It uses 2 organism images of the same coordinates and compares them by using a specific algorithm called the identification procedure. This algorithm assumes that the spot pattern of each individual is a unique and distinguishing feature. The user points out the most distinguishing spots of each image. The spot pattern is stored in what is called a fingerprint file. In the identification step, the resulting spot pattern is matched with the spot patterns of all the known animals in the database. The user is always required to point out 3 fixed reference points. These points must be visible in all images, clearly distinguishable, and the triangle created must cover most spots. A comparison of the 2 images comes down to finding the corresponding spot pairs. The red dot denotes the spots of the first image, while the blue circles represent the spots of the second image. The green lines indicate whether 2 spots are considered a matching spot pair. From these pairs, the distance metric is calculated to be able to rank each image in the database. The current metric is the sum of the distance between each spot pair divided by the square of the number of spot pairs. Differences in shape and size will also affect the final score. This picture shows a comparison of spots of the 2 images. The blue dots indicate similarities. Another program that can be used to identify organisms is the Sci Res program. The program can be used for identifying patterns on the chest, abdomen, and neck. With minor modifications, it can be also used for seal patterns. The program fits a 3D surface model to the image, and the program captures a pattern that is unaffected by the camera angle or posture. The yellow dots help achieve the adequate fit needed. After extraction, the software goes on to compare the new pattern with previous patterns stored in the library and displayed the most likely matches. The resulting database of match results can be used for monitoring of population size and other parameters, determining the fate of individual animals, encouraging cooperation between different research groups, and so on. Another program is the MATLAB developmental system. The image appears in a new window next to a provided standard image, which is simply an image of excellent quality used as a reference image to help map features on the new image to be entered into the system. The program scales, rotates, and translates the new image so that all images have the same rotation and scaling. The normalization parameters are saved and are applied to each image each time it is read by the program. Because some regions of the organism are not clearly visible on some photographs due to wash, glare, and etc., the user may designate all parts of each image as a region of interest for matching. David Rotet studied population abundance and apparent survival of the vulnerable whale shark Rhinodon typus in the Seychelles aggregation. He evaluated photographic records over 7 years of vulnerable whale sharks for estimations of population size and structure. He used Sci Manta for comparing individual spot patterns behind the gills, as well as marker tags on the sharks. The combination of photo identification with marker tagging identified a total of 512 individual sharks over 2001 to 2007. There are 115 resettings in subsequent years. His results find that there is a transient population of whale sharks in the Seychelles, and that regional conservation approaches are required. Another researcher is Kelly Hastings, who investigated the use of computer-assisted matching of photographs to examine population parameters of Alaskan harbor seals. She used the Sci Res program as the photo identification software for matching photographs to examine harbor seals. She took photos of seals in Tugdeg, Alaska from 1998 to 1999 and categorized photos into pups, yearlings, subadults, and adults. She then used the Sci Res software to determine fingerprint regions of the side of the head and the front of the head. She found that the matching software performed well when used with the side view, but poor when used during the front view, with a 6.4 error rate for the side and 34% error rate for the front. Her data can be used for estimations of population parameters. Another researcher is Gilbert Hillman, who investigated photo identification software for bowhead whale images. He developed a program for identification of whales in aerial photographs using MATLAB. The program enables the user to create a collection of images and compare an image to the collection. The collection is ranked by the program based on similarity to the query image. The program was tested by the developer with 20 images and had a 2% error rate. 11 successive versions were tested with a collection of 78 images. Additional modifications to the program are likely to improve matching performance when larger numbers of images have been processed through the program. Based on the work of David Rothart, Kelly Hastings, and Gilbert Hillman, the hypothesis of the study is that Sci Res will be most effective at identifying harbor seals, and MATLAB and I3S Manta will be least effective respectively. Images of the seals will first be collected in the archive folders of Dr. Arthur Kopelman photos and used until early December when seal migrations start. During seal migrations, photos will be taken at the haul-out pattern of Cupsock Beach, New York, and continue until early May. Photos will be taken of the side of the head as well as the front of the head. All photos will be categorized into sex, class, age, which if the seal shows its ventrum, color phase, light, medium, and dark, and conditions of the pelage, dry or wet. To assess the accuracy of the identification techniques, there will be calculations of the two error metrics common to biometric recognition studies, false rejection rates or FRR, and false acceptance rates, FAR. FRR is defined as the frequency of failing to match two captures of the same individual. It's the amount of false rejections divided by the amount of identification attempts. FAR is the frequency of falsely matching two captures of different individuals and is calculated as the amount of false acceptances over the amount of identification attempts. All softwares are free of use. The laptop must be able to run Java 1.5, which is attainable at home. The Canon EOD 70 Wildlife camera is attainable also at home. School supplies are readily attainable at Manhasset High School. The only thing needed is a Pinniped pass for unlimited seal walks, which is $30. Thanks for watching.